This method can help answer key questions in the cancer metastasis field about the role of lung colonization in accomplishing cancer metastasis. The main advantage of this technique is that early extravasated cancer cells can be visualized in the lungs during cancer metastasis.
Demonstrating the procedure will be Cheng-Han, a PhD student from my laboratory.
When the tumor cell culture has reached 70 to 80% confluence, wash the culture 2 times in 2 milliliters of sterile PBS per wash and add 1 milliliter of 0.05% trypsin EDTA to the culture dish. Immediately remove 800 microliters of the supernatant and place the culture dish at 37 degrees Celsius for 30 to 60 seconds until the majority of the cells have detached from the plate. Add 1 milliliter of fresh medium supplemented with 10% FBS to stop the reaction, and use a 1000 microliter pipette to vigorously mix the cell solution. Transfer the resulting single-cell suspension into a 1.5 milliliter microcentrifuge tube and collect the cells by centrifugation. Then resuspend the tumor cell pellet in 1.5 milliliters of medium supplemented with 20% FBS and end-over-end rotate the cells for 2 hours at 37 degrees Celsius.